back to my channel and happy Friday. My name is Jess if you're new to the channel, but today's video is going to be a pretty interesting one just because it's going to be really about my summer research program, and I'm going to take you guys along in a day of like what we usually do. It changes and it varies, of course, because it depends on what we're extracting, what we're looking at, and what we're sorting. And today is going to be more of a DNA extraction according to soil. So we took some soil samples. We went to ABK, which is the site we usually take a lot of our samples in. And yeah, so we're going to learn how to do a soil extraction DNA. So I thought it would be fun to kind of vlog it and see for you biology majors out there or future biology majors. Maybe you guys can stumble upon this video and see if you guys are gonna like the lifestyle of being a biologist. So basically we do a lot of in-lab activities, and you're really gonna be in lab like for the next 4 years of your college career. So get used to it, love it, and learn it. So yeah, it's gonna be fun. Noise. Okay, we had 6 samples. Dang, I can't even see the, oh wait. I can't even see the writing on that one.
Oh, label the bags again.
Just rewrite it again?
Yeah.
These are all first samples that we took from the field a while back ago. All nicely labeled.
Okay, what's next? Okay, let's get the tubes out and
This
see
one?
if, yeah, or all of them. And then let's see which ones we're going to be using. And also, let's get our tube rags. This one is different. I don't know why we were using that one, because that's old. This, we just go through the steps.
As like we would do to that one.
Yes.
Okay.
So we'll put it, well, actually, we'll just leave it here.
Yeah.
Change my gloves. So,
There's so many instructions. But this is how we're gonna sample the soils. Instead of plants, we're doing soils today.
let's read through here and find out which ones we need.
Okay. Notes before starting. Ensure that the power feed pro tubes rotate freely in the centrifuge without rubbing. If solution 3D3 has precipitated heat at 60 Celsius and temperature dissolves, perform all centrifuge steps at room temperature. Spin the power bead pro tube briefly to ensure the beads have settled at the bottom. So we need beads. Add up to 250 mg of soil and 300 units of solution with CD1 vortex briefly to mix. Secure the power bead pro tube horizontally on a vortex adapter for 1.5 to 2 ml tubes. So we need 2 ml tubes. Vortex at maximum speed for 10 minutes.
Yep.
If using the vortex adapter for more than 12 preps, increase the vortexing time by 5 to 10 minutes. For more information about your bead beating method, see the protocol detailed section. Centrifuge the power bead pro tube at 15,000, okay, and then transfer to a clean 2 ml. So we need another clean 2 ml microcentrifuge tube. What's the difference? One is just regular 2 ml tubes. Okay, for centrifuge tube.
Oh, these are beads. I thought this was powder.
Oh, they're different. They're different, guys. Instead of it looking like little rocks, they're white this time. How do you remember this? This is yes. And we're vlogging.
And you're vlogging?
Yes.
Oh, cool.
I bought a tripod.
That's so awesome. How many times I wish like I could do that during my labs.
Centrifuge. What's the difference between 2 ml collection tube to a 2 ml micro centrifuge tube?
So the collection tube
Is the one with the little like, what is it called? Where it catches it, like a little filter?
or no, this is
Oh no, collection tube is
collection
just
tube.
regular.
This is your spin column,
Spin column.
or the thing that's in there. It already comes with the collection tube, and then this is a
Regular 2 ml.
elution tube.
I did not see that on there. Do we have a 2 ml
Oh,
micro centrifuge?
Yes, we have 2 ml centrifuge tubes as well. Alright.
Okay, okay.
Then we have. Oh, alright.
You said we have to disinfect the scale with bleach.
Yes. So.
We're gonna be cleaning the scale. So there's no contaminants because we use it for grass too. And now we're using soil, so we're gonna clean it up. Just finished, well, I just finished wiping that down and wiping our table down just to make sure there's no leftover DNA samples from anything else. Okay, so now we have 6 weigh boats that we're going to be using. I'm going to have to disinfect it with bleach and ethanol just to make sure that it is properly cleaned and no contaminants, no DNA, just in case somebody ended up using these. But let me get the bleach and the ethanol real quick. So bleach first. I just bleach with the paper towel just so it's not messy and getting it all over the place. But I'm just gonna clean it thorough. I'm done. I'll probably put some in. Actually, maybe it's more effective. And so there's a fresh set of bleach in each one. Looks about good. Yep. So today should hopefully not be too long. I think we only have this planned, and that's it, which is just soil, DNA extraction roofing it, and that should be it. We did a lot of our plant samples already, so, and the only other thing that we might be doing maybe is insect sorting, which might be a possibility since we haven't been in lab in like a good week since summer 2 classes started, so we're trying to get like set up in with that. Next is just gonna be 70% ethanol, and it's for cleaning. So I'll just put some in here as well. And I have another paper towel ready to use. But yeah, so it should be a pretty chill day. I was actually at work from 7:30 to 10:30, and for the rest of the day, hopefully we're not here past 3. So 10:30 to 3 in lab. Because because yeah, we wanted to get an early start because last time Danielle and I were here till like 7. I don't want to go home at 7 p.m. today, but we'll see. It just really depends if we decide on doing insect sorting. We wanted to take pictures and try to identify a lot of them already, and so we can kind of put it in her journal and her presentation, because I know she's making a poster board that has all the information of like her research study. So yeah, so I guess we'll see where the day takes us, but it looks like DNA extraction for the soil is gonna be a while. There's a lot of like steps to it, and it's like 10 minutes for each spin cycle, which is a lot. So give or take probably like 2 hours of like DNA extraction by itself. But yeah, so all of these are clean. I'm gonna wait for it to dry a little bit. It should dry quickly because of the ethanol, and then we're gonna weigh the soil, and we need 250 grams, I believe, of each sample sites. But yeah, Danny, I'm just gonna label the weigh boats according to the soil.
Okay. Actually,
What's up?
let's do S1 through S6, and let's assign the soil bags to an S1 and S6. And let's go ahead and write that down.
Okay.
And notebook.
S1. So how do you want to determine if they're S1?
Let's just do it in order of
Meter point?
Yeah.
So 27, 31, 32, 38, 43. 1, 2, 3, 4, 5, 06. So this is in order.
Right here, so that way we can turn back and look. And if you want, you can take a picture of it too.
Probably be the easiest.
Mm-hmm.
So this is 1, 2, 3, 4, 5, 6. Okay guys, so now since I was able to label each weigh boat according to each sample, there's only 6 samples, but now I'm gonna have to weigh about 2.25 grams for each one. So I'm gonna wait till that weighs out, and then our first one's gonna be from site number 1 at 11 meters, and it's gonna be for silver blue stem. And okay, so now let's sort that out. Take this. Let's try about this much of soil and see how much that is. It's not a lot. So for 2.6 is that much soil. Can you guys see that? It's a little bit of soil. Let me ask Danny to make sure this is an appropriate amount of soil, because I've never done soil before. But where did Danny go?
Oh, was you talking about that guy outside, that older guy? Not the older guy.
Is it Watson?
I think she said it was Watson, but I think she said something about his son. Is his son here?
I have no idea. I've never met his kids.
They do have the kids or the young people doing the
Oh.
tour, so he might be part of the tour group.
I think Okay. I think got her attention and now she's shooting out.
Yeah, they're supposed to be incoming freshmen.
What?
Well, like, no, not college freshmen, but actually like high school freshmen.
Oh, just like tour the campus?
They're doing what are they doing?
Or is it kind of like
They're
a...
doing like a program, like a summer program.
Okay.
So on Monday, we did like little events with them.
Oh,
so, like,
cool.
little sections, like
Yeah.
there's like a CPR section.
Oh,
What?
yeah. CPR is in like the first aid class.
Yeah. So we were teaching them how to do that.
Wow.
Yeah. So there's like different sections that they're doing.
That's cool.
Yeah. Sample, but I just want to make sure this is like.
That's 0.25.
0.26.
Yeah. That's alright.
Okay, so she agreed with the amount because I was kind of worried about how tiny it is, but she said it's okay. And then now, since I use this to sort out the soil, I have to disinfect it. So I'm just gonna dip it in ethanol, and we have like this little small fire thing where I can light it, and then go over. That should be good. It's just to kill off the other sample DNAs and not get cross-contamination. But let's get started on this. Too much. That's too much. I'm really trying to just grab a little time. Which has to do, I know. This is so much easier. So like grab. We're working out what we have, guys. Awesome. So now we're at 0.26 grams, which is again this much. Take this leaf out, just about this much, and then we're gonna put them with the beads that you guys saw earlier. I hope that's focusing correctly, and we're just gonna insert some of these in, well, all of these in here.
What it looks like, and we'll get everything above the soil or whatever.
What happened to the beads?
They're in there.
They got mixed, like they're just mixed into the soil.
Yeah, I see them.
Interesting.
They're small. They're so tiny.
You know that little trick you do, Danielle? Can I do it with this one? Or no?
Yes. A trick. So
We don't have to keep changing the tip.
Instead of changing the tip, just
Do it right above it.
yeah, do it right above it, and
Solution
then
CD2.
be very and be careful.
Yes. Like we really have to like just stay.
And then if you do feel like I got it, and just change it.
Change the tip. Okay.
But we're trying to save time
Trying to
and resources.
And resources. There's
Yeah.
too
Is this
much-
set already?
No.
You said 200, right?
200. Yeah.
Oh, you could use the other one if you want it to be more precise.
Yeah. I was
Can
like,
you
this
set it
one
to
could
200, please?
There's too much plastic.
Yeah. Too much plastic waste.
We're killing the earth,
Yes.
and we're studying the earth.
Which one?
And I didn't know it existed. True Blood.
Oh,
True Blood?
that's on Netflix, right?
That
Or am I
vampire
tripping?
show? Yes. HBO Max, I think.
Yeah.
Because we're taking pictures.
We're taking pictures, but also some of the insects we got from T2,
Yeah.
I don't have anymore.
Why?
Because I sent them to Baltimore.
Oh,
Like, I know for sure we had a tree hopper in there.
okay.
And I sent that to Baltimore. So the ones that we really be working on is,
T1.
yeah, T1.
Okay. Okay, we can.
And then all of them are separated by species already.
Yeah. So are we just taking pictures?
Yeah, we would just be taking pictures at this point.
Okay. So we're gonna be separating some of the insects from Transic 1, which is site number 1. and from previous times we already separated it by species, so these should all be the same. Then these are tinier. Oh no, this one's a little open. Tinier ones. Nice.
Look at those little hairs.
Danielle's trying to fix up the camera with the
You
microscope.
can't see it all like on one take though.
No,
you can't really.
You see the little
This would be
bug?
maybe a good example of like a video.
Hey guys, so we made it back to my car and decided to end lab a little earlier than usual. So we finished doing the DNA extraction of the soil, and that took like about 2.5 hours to do like the whole entire process of separating and doing all like the 6 samples, which is fine because it's kind of it's a simple work, but it's just a lot of steps to it. And then it was kind of fun. I haven't seen Danielle and Sarah in over like a week and a half, just because summer 2 classes started, like I said. But yeah, but I'll see them tomorrow at 9, and we're gonna just take pictures of all the insects and probably separate a little bit of the leftover ones that we missed. We swore like that we finished everything, but I guess we missed like 2 jars, so we're gonna separate those, which isn't too bad because there wasn't a lot of insects in them, and then just take lots of pictures and try to figure out their actual proper name species. So yeah, shouldn't hopefully be too long, fingers crossed. But I just really wanted to end this vlog, and I hope you guys like this type of content, and if you guys do, give it a thumbs up or write a comment, and maybe I can get more into like focusing on my biology and like what I do, and then maybe the classes I start taking in the fall. This is my senior year, but it's okay. I can definitely try and explain more about my experience as a biology major and like what I've been doing. But yeah, I hope you guys like this video. Please like and subscribe, and I'll see you guys in my next one.